Okay, good morning. We're going to get started now, and we're going to take a look at metabolism. And we started on that last Thursday. It seems like forever ago, but that's where we're going to start with that and work our way through and see how far we can get. I definitely know that we're going to wrap up this chapter, not today, but definitely Tuesday. And we'll be able to start on the bone chapter, looking at the physiology of bones. And I will tell you that lecture content gets a whole lot more interesting from here on out. This content is somewhat tedious, but I will be trying to explain the practical applications for you. And for some of this stuff, no one's ever going to ask you about, but it does give you a good understanding of how the human body functions. And I'll try to explain that as we go along. So anyway, moving on, let's go ahead and get into chapter. I guess it would have worked about that. Back here and start going backwards. And don't mind me while I talk to you. So I'm going to start here on metabolism. I guess everybody can see this now. You can't see it. I'm sure someone will tell me, and otherwise I'll just ramble on without any pictures. Metabolism is the sum of all the chemical operations going on in our bodies. In fact, I could probably go back to this point. All of the reactions going on inside our bodies, they are either going to be an anabolic reaction, an anabolic chemical reaction, or a catabolic reaction. Anabolic reactions when we take little molecules and make big ones, like we take amino acids and we make proteins. We take glucose and we make glycogen with it. We take chopped up fats and we make triglycerides with them. So we make anabolic reactions, take little things and make big things out of them. And it always takes energy to do that. Anabolic reactions take energy. It's like baking a cake. You can mix all the ingredients together, but you're not going to get a cake unless you put it in an oven and raise the temperature. Not going to work. You have to apply energy to it. Now, catabolic reactions are the breakdown of large molecules into small ones. We eat protein, and we break the protein down to amino acids. So then we can use the amino acids later. We take carbohydrates, starches, and break them down to glucose. Glucose is what we use for fuel. We take triglycerides and break them down into smaller pieces of fat and then reassemble them back into triglycerides. So catabolic reactions take large molecules and break them down to small ones, and they always release energy. Because when you're taking a large molecule and breaking it down into smaller ones, you're going to end up breaking the covalent bonds that hold that molecule together. And when you break those covalent bonds, you're releasing energy. So anabolic reactions require energy and they build things up. Catabolic reactions give off energy and they tear things down. After you bake the cake, you eat the cake, or somebody eats the cake. And there's a catabolic reaction involved there because you eat the cake and you break the complex carbohydrates down in glucose and you release energy. So anabolic and catabolic reactions. Oops, I went the wrong. Sorry. And I just said all that. Anabolic reactions give us what we need to build proteins, to build up glycogen, to build up fats for maintenance and repair in our bodies. We make proteins, for example. Those are anabolic reactions. Little molecules are strung together to make big ones. Glucose makes glycogen, for example. And catabolic reactions are when we break things down. We break down fats into its individual components. We break down proteins to amino acids. We break down carbohydrates to glucose. Those are catabolic reactions, and they give off energy when they do so. When you light a match, that's a catabolic reaction. You're releasing energy stored in the match, whether it's in a wooden match or a paper match. You know, the match head ignites and causes the match to burn. You're releasing the energy there. You're taking a large molecule that, like a wooden match, the cellulose and lignin in the wooden matchstick, and you are converting that to carbon dioxide and water. So you go from a large molecule down to a little one, and you release energy. In our bodies, we use catabolic reactions to break down the food that we eat and release the energy stored in the food molecules, and we use that to form ATP. We can't take the energy that's found in food and use it directly. You know, you look at a Snickers bar and it says 180 calories. That's a measure of all the potential energy stored in that Snickers bar. You know it's potential energy because the Snickers bar isn't moving, but it's the potential energy. And that potential energy comes from all the electrons in all the covalent bonds of all the carbons and hydrogens and oxygens in the glucose found inside the Snickers bar. You know, it's all the carbohydrates and all the fats and all the protein, because there's protein in the Snickers bar. The peanuts give you protein. But it is not, never mind, I won't go there. We break down all that potential energy. We break down the complex molecules in the Snickers bar or the apple or the banana or the carrot or the potato or the biscuit, you know, or whatever. We break down those large molecules into simple ones and we release energy. And we can't use that energy coming directly out of the food because we can't deal with it. We have to park it in something that we can some vehicle that we can use it in, and that's where ATP comes into play. ATP can grab little bits of energy. 7.3 kilocalories of energy. 7.3 kilocalories of energy in each ATP molecule, and that will last for 7 seconds. And so for 7 seconds, we can transport that ATP around to where it needs to go. And we can do things with that ATP molecule. Because we can take the phosphate off the ATP and transfer it to a structure, and that the phosphate on that third phosphate on the ATP has the energy attached to it or embedded in it. And when that third phosphate binds with a structure, it's now energized. Like the sodium potassium pump uses the third phosphate to power it. Our skeletal muscle cells use the third phosphate to power a muscle contraction or for a nerve impulse. So we use the ATP molecule as the currency, as our money to get things done. we use the energy taken from food to make ATP. We shift the phosphate, the third phosphate of the ATP molecule, to other structures to get things done. And those, once that third phosphate is attached to the sodium potassium pump, the pump will work. We say it's phosphorylated. Nice way of saying that the phosphate's attached to it, at least temporarily, for 7 seconds or whatever. Yeah, so the process of phosphorylation is lending that phosphate, that high energy phosphate, to the structure that needs it for a few seconds. So it can do something, so it can pump sodiums out, so it can cause a muscle fiber to contract. So, and this is how we take the energy from our food that we eat, the apple, the banana, the Snickers bar, the steak, the potato, the chicken, the fish, the glass of milk, the bread, the roll, whatever. All the calories in there represent the potential energy in all those foods. And that potential energy can be channeled into the production of ATP. We can release that energy from the food, and that can be channeled to make ATP. And then we can use the ATP as our method of distributing that energy throughout our bodies. That's how we do it. Now, we use enzymes to control this. We certainly don't allow the random production of ATP molecules all the time. We certainly don't want to have a lot of energy released all at once either, because that would be very bad. There's a tremendous amount of energy in a molecule of glucose, and we certainly don't want to release that at one time. We have to have a step-by-step controlled release, and we use enzymes for this. Enzymes, of course, are the biological catalyst. That's the definition of an enzyme. A biological catalyst that lowers the the activation energy needs to start the reaction. That's great. That's the textbook definition. So what do they do here? Well, they can break down starches to glucose. They also can take glucose and strip off electrons by stripping off hydrogens. There are enzymes called dehydrogenase enzymes. Remember hydrogenation? Hydrogenation is the process of adding hydrogens into oils to make them solid at room temperature. So sort of breaking the rules, if you will. Dehydrogenation is the process of removing hydrogens. Now, we usually don't do that to food, but in our bodies, the food industry doesn't take hydrogens out of food. They put hydrogens in. But in our bodies, we want to strip off hydrogens from food. We use enzymes called dehydrogenase enzymes. Those enzymes, when they strip off hydrogens from like glucose, for example, or a starch or a fat or a protein, when they strip off hydrogens, hydrogens don't travel alone. Hydrogens are protons. They have a positive charge. The hydrogen ion is a positive charge, also known as we call it a proton. We just say hydrogen ions. But their best friend in the whole universe is the electron. And so the hydrogen ion will always travel with an electron. So if you pull a hydrogen off of a molecule, you're going to get an electron to come with it. And the electron contains energy, potential energy, that you can do things with. So our enzymes can control the rate of chemical reactions of metabolic reactions by how rapidly we can allow ATPs to get made, how rapidly we're going to break down glucose, or break down sucrose to glucose, or starch to sucrose to glucose, or fats to glucose, or proteins to glucose. our enzymes are very specific. They are substrate specific. Remember what the substrate is. The substrates are the reactants. They come together, they land on the active site. The active site of an enzyme is that very specifically shaped structure that only a unique substrate can land on. The enzyme amylase in our mouth breaks starches down into smaller molecules. And it's designed, the only thing that will fit on the active sites of amylase are glucose-shaped molecules. That's all. Nothing else can land there. Remember, what is a starch? A starch is made of a whole bunch of glucoses linked together. So the active sites of the amylase are shaped to receive glucose held together, and that's all that fits there. Nothing else can land there. That's the only thing amylase works on is starch. And it can break the starch into smaller and smaller pieces till we get glucose. And the only place amylase works is in our mouth. The active site, the shape of that active site determines what substrate can land there. All the other things that are in our mouth when we're chewing up food, that's where digestion starts, by the way, are going to try to land on that, on the amylase, but only starch fits there. When we eat food, we have to break down the nutrients. We have, the first step is digestion, absorption, and transport. Digestion starts in our mouth. We start chewing up foods. The saliva contains all sorts of enzymes. Amylase is found in our saliva. Our salivary enzymes start the process of digestion. It continues in our stomach. It continues in the first 12 inches of our duodenum, the first part of our small intestine. And we finally break everything down to where it can be absorbed. And absorption starts in the next section of the small intestine after the duodenum. The duodenum is only about a foot long. Duodenum is where we finish the process of digestion. We start absorbing nutrients then in the next section called the jejunum. And we'll get into all that in AP2. You know, and the small intestine is about 20 feet long, about the size of big around as your finger. And it's mostly lined with simple columnar cells, and it absorbs our nutrients. And once the nutrients go through the simple columnar cells, it either enters the capillaries or it enters the lymphatic vessels, and fats go in the lymphatic vessels, and sugars and proteins go in the capillaries, and they all end up in our blood supply. And once they're there and they go through the liver, they go out to all the cells. So that's step one. Once we get into the cells, and every cell is in contact with a at least one capillary. Once it gets to the cells, we start processing the nutrients in the cytoplasm. Glucose, we start to break down of glucose in the cytoplasm. Proteins are assembled in the amino acids from the proteins are assembled in the cytoplasm in the rough ER. The fats are reassembled in the cytoplasm. Remember, because we break down fat into a monoglyceride, which is one fatty acid chain attached glycerol. That's a monoglyceride. And two random loose fatty acids. And we reassemble them into triglycerides inside the cytoplasm. That's the first step. We start, we put the lipids back together again. We put the we convert amino acids into proteins, and we can take glucose and make glycogen. If we have lots of glucose, we can make glycogen. If we have a lot more glucose, we can convert it to fat in the adipose cells. Now, in the cytoplasm, we will take some of that glucose and start breaking it down through a process known as glycolysis. We'll start will continue the processing of these nutrients, the breaking down of these nutrients. We'll start with glucose, because glucose is what we use, but we will start breaking down glucose in the cytoplasm. And we'll get a little bit of a payoff there. The last step takes place inside the mitochondria, because the mitochondria is the conversion center, the energy conversion center. And we get our big payoff there. We produce a lot of ATP, but we'll also release carbon dioxide and we'll release water. But this step in the mitochondria actually requires oxygen. Nothing to this point, nothing to step 3 actually required oxygen. Oxygen gets used in step 3. What we start doing in the cytoplasm doesn't require oxygen at all. So for those of us that are more visual, this shows you what's happening, what I just explained. Here we are, we have eaten protein and carbs and fats, and we get, by the time we get to the duodenum, the first 12 inches of the small intestine, we have broken our proteins down. We finished breaking proteins down to amino acids. The carbohydrates are broken down to glucose, and the fats have been broken down to the fatty acids and glycerol, or glycerol with one fatty acid attached to it. Once we get into the cells, though, the amino acids are converted back to, are made into the proteins we want. It's the same amino acids, there are only 20 of them. Over here with the fats, the glycerol and the fatty acids are reconverted inside the cells back to the triglycerides, the neutral fats. And our proteins are used to whatever we're going to use them for, and the fats are going to be used for good or for bad, whatever we're going to use them for. But the carbohydrates are converted to glucose, and glucose can form glycogen for storage, or glucose can undergo a process known as glycolysis. We start the breakdown of glucose to release energy called glycolysis. And that takes place in the cytoplasm, nowhere else. The end result of glycolysis is a substance known as pyruvic acid. And pyruvic acid can enter the mitochondria. We go into the third step, where we actually release lots of energy. We have something called the Krebs Cycle and the electron transport chain, where we get a big payoff on energy. Lots and lots of ATPs get formed. We know what energy does for us. Energy is the ability to do things, to do work. Well, there are a lot of energy forms that apply in our bodies. Heat, we give off a lot of body heat. 60% of the energy we obtain from the food that we eat radiates off of our bodies as heat. 60%, that's a lot. But it means we have 40% of the energy in food to do things, and that's a lot too. We depend on light energy to see things. Photoreceptors in our eyes receive light rays. We also depend on light energy for producing vitamin D in our skin. Sound energy, sound waves, stimulate our auditory nerves so we can hear things. We also produce sounds. We have a heartbeat. We have a heartbeat that occurs every 7/10 of a second. That's the sound of the valves closing. That loved up sound that you are going to hear all about in AP2. This is a sound that we make in addition to being able to hear things from vibrations. We generate sounds. We can listen to our heart. We can listen to our lungs. We can listen to the sounds that our lungs make. We can listen to the sounds that our digestive system makes. Electricity, we are electrical. We are charged. We have minus 90 millivolts on our muscle cells and minus 70 millivolt charge on our nerve cells. And that powers our activity. We generate electricity in our cells. We produce mechanical energy by moving things and contracting muscles and moving our bones. And we use and store chemical energy all the time. We park chemical energy in the form of glucose as glycogen. We park triglycerides in the form of fat in adipose tissue. So we are transferring ATP molecules and using the high energy third phosphate to do things. So we use lots of energy in various forms. And every time we transform energy from one form to another, a little bit of that energy is lost as heat. When we go from chemical to mechanical, we lose a little bit of that energy as heat. We don't destroy it. It just moves into a form that we can't do anything with. You know, when heat radiates out from our bodies, we can't capture that energy and do something with it. All metabolic activities involve these various types of energy. And ultimately, we're going to end up losing energy, losing its use as heat. Everything revolves around this heat energy we can't get back. But because we have 40% of the energy that we take in, we do things, we get things done. So now, most of what we do is based on chemical energy. Most of what we do is based on chemical energy. The energy that we draw from the foods that we eat, transferred around our bodies. You know, we can't, our photoreceptors can't function without chemical energy. Even though light energy is stimulating them, our photoreceptors in our eyes can't function without chemical energy. The same applies to the auditory components of our ears. The stimulus that we receive, the mechanical energy that's in the sound waves, the vibrations in the sound waves are converted ultimately to nerve impulses through chemical energy. So everything, nearly everything is going to be chemical energy in nature. Now, most of the chemical energy that we use comes from the oxidation of glucose. Most of it is from the oxidation of glucose. And some of it is, well, I'll explain that in a second. So let's say most of the energy is coming from the oxidation of glucose. Oxidation is when you burn something, when you strip away electrons, when you release energy. That's oxidation. When you're releasing electrons, you release energy. That's oxidation. When something is oxidized, it gives up electrons and releases energy. When you light a match, when you have a fire in a fire pit, when you burn gasoline or diesel to drive somewhere, you are releasing the stored energy in there through the process of oxidation. You're burning it. You're burning the energy, burning the fuel in your fire pit, your fireplace, your charcoal grill, your gas grill, in your engine, in our bodies. You are burning the fuel and releasing that energy. And cellular respiration is the term we use to describe not breathing, but the conversion of chemical energy stored in fuel, the 180 calories in a Snickers bar, converting that stored energy into something that we can use to move our muscles or pump our blood or whatever. Now, the term cellular respiration, as I said, does not have anything to do with breathing. Breathing is, we have external and internal respiration. We'll talk all about that in AP2. Cellular respiration is a different process. It has nothing to do directly with breathing. There are three parts to cellular respiration. You have what's known as glycolysis, which make a note to yourself on your printout, in the cytoplasm only. Glycolysis is the only step that takes place in the cytoplasm only, nowhere else. None of these other steps take place in the cytoplasm. So glycolysis, followed by the citric acid cycle, or most of us refer to it as the Krebs cycle, because Krebs was a biochemist that discovered this, probably 100 years ago. And then the third part is what's known as the electron transport chain, because we're going to be dealing with electrons, high energy electrons that have all that potential energy. These are the electrons that we're stripping off of glucose. They were the valence shell electrons, and valence shell electrons have lots of potential energy. That's what we're going to be taking advantage of. The end result of cellular respiration is going to be the production of carbon dioxide, the CO2 that we breathe out all the time. That CO2 is coming from the mitochondria of the cells, and only the mitochondria, nowhere else. The production of water, the generation of ATP, parking chemical energy in the ATP molecule, and the release of heat. Remember, 60% of the energy that we take in comes back out as body heat. These reactions can occur both anaerobically, meaning without oxygen. The term aerobic means involving the use of oxygen. Anaerobic, the AN in front of that, means that there is no oxygen used. We have both types of chemical reactions in our bodies, anaerobic and aerobic. Anaerobic reactions don't require oxygen, but only give you a little ATP. Aerobic reactions have to have oxygen and give us a boatload, that's a technical term, a boatload of ATP. And the process we follow to do this is a series of reactions called oxidation reduction reactions. Oxidation reactions give up electrons. I'm sorry. Let me say this first, then I'll say give up electrons. Oxidation reactions cause molecules to lose hydrogens. When something is oxidized, it loses electrons. And when an electron goes, its best friend in the whole universe, a hydrogen, goes with it. Because electrons are negatively charged and hydrogen ions are positively charged, and so they travel together. The universe doesn't like us to be unbalanced. So we have the positive hydrogen ions and negative electrons, and if we lose an electron, if an electron is stripped off of a glucose molecule, a hydrogen has to come with it to keep balanced. So when something is oxidized, when you are burning a candle, you light a candle, and you've got the flame, and you've got the heat and light coming off of that, and you can see the wax getting smaller and smaller as the candle burns. The candle is giving up electrons in the covalent bonds of the paraffin, the wax that the candle's made of. You light the candle, and the flame from on the wick is causing the covalent bonds in the wax to break apart, and that releases energy. That energy shows up as heat and light. The flame continues to burn here. So you're releasing electrons. Those electrons are being released with hydrogens. And those electrons and hydrogens are coming off the wax. But that's only half the battle. While the universe doesn't like to let electrons travel by themselves, or hydrogen ions to travel by themselves for that matter, because they have to travel in pairs of a positive charge and a negative charge, they have to go somewhere. When they get released, when we break apart a glucose molecule or burn a candle or light a match or burn gasoline, the electrons that are getting released to generate with the energy that's, when you're burning the fuel to release the electrons and release the energy, because that's what it's all about. When you break the covalent bonds and free the electrons, you free the energy too. But those electrons and hydrogens have to go somewhere. The process of oxidation is stripping away electrons and hydrogens. The process of reduction means that every time you strip away an electron, something else has to grab it. Every time you strip away a hydrogen, something else has to grab it. And that something else is usually oxygen. Remember good old oxygen. It has an atomic number of 8. 2 electrons in its first shell, 6 electrons in its second shell, room for 2 more. And it's always hungry for electrons. Well, if it sees 2 random electrons coming off of glucose, it's going to grab them. And it's going to pull the hydrogens with it. And every time you do that, you get a water molecule. So when something gets reduced, it's going to gain hydrogens and electrons. The substance that gets reduced is usually oxygen. Only a chemist would say that when something gains electrons, is it going to get reduced. But that's neither here nor there. The point is, though, that we don't have oxidation unless something can grab those electrons coming off. You can't burn a candle in a vacuum. You can't burn anything in space. There's no oxygen there to grab the electrons. Electrons have to go from the substance that's being burned to oxygen. You're going to release carbon dioxide and water in the process. We call these things redox reactions. The substance that is being destroyed, the substance that's being burned, the substance that's being oxidized. Now, here's where it gets confusing. Something that's being oxidized is the reducing agent. Why? Because it's going to generate electrons. When something is being oxidized, it's releasing electrons. That makes it the reducing agent. This is chemist talk now. The substance that accepts the electrons is the oxidizing agent. Oxygen is nearly always the oxidizing agent. It's an easy way to remember that. Because we can't burn something without oxygen. We've got to have oxygen to grab the electrons. If we can't grab the electrons and the hydrogens, then nothing's gonna happen. You can't, with no oxygen around in space, you can't strike a match because there's no place for the electrons and the hydrogens to go from burning the wood or light a candle. You can't. There is no oxygen present in here. So when something is oxidized, it's going to get weaker. It's going to give up electrons because the covalent bonds are being broken, and it's going to give up its hydrogens. Oxidation of food ends up, we release energy from those covalent bonds, and we produce carbon dioxide, and we produce water, and we make a lot of ATP. We burn gasoline in our car engine, and we give off heat, and we give off water vapor through the exhaust and carbon dioxide, and we convert that chemical energy into mechanical energy because we're breaking those bonds. And all that chemical energy gets released as mechanical energy, and we power our vehicle. But the end result is you can't run at a gasoline or diesel engine in a vacuum. Now this little illustration here, probably, I don't know if it's more confusing to look at it or not, but if you consider that this is our glucose up here, the compound A is glucose. It's going to give up its electrons when it's oxidized. Now in its oxidized state, it has lost its electrons. Or you've burned the candle, you burned the wood, and there's nothing left. You can't oxidize compound A unless there's something here to grab the electrons. So compound B is the oxidizing agent. That is oxygen. Compound A is the reducing agent. It generates the electrons for you. So oxidation and reduction. Why do we care? Because the whole process of getting energy out of our food depends on stripping away electrons from the food and transferring those electrons somewhere else. Because we can't just let them run randomly. We also can't release all the energy at once in our cells. That would be really bad. There's a lot of energy in a molecule of glucose. And if we were to release all that energy in our cells at one time, it would be very, very exciting, at least briefly exciting. Now, we don't want to do that. We have to we have to break the glucose down slowly and go it through a step-by-step process. Yes, we want oxidation. We want to strip the electrons off of our glucose molecule and its best friend hydrogens to come with it. But we want to do it slowly. We don't want to release it all at one time. And we can't do it unless there's something there to grab those electrons. If we can't grab the electrons coming off of glucose, we're not going to have any oxidation at all. If we can't grab the electrons coming off, they're not going to come off. So when we lose, bottom line then is when we lose electrons, it's oxidation. We lose electrons, we lose energy, we lose hydrogens. Something has to accept those electrons, and that something is then reduced. The process of reduction. Chemists call it reduction because you are reducing the number of positive charges in the molecule that's accepting the electrons. Makes perfect sense to me, Most of us think reduction means that something shrinks. Well, not in the case of chemists. Chemists will say that we have reduced the number of positive charges, we've added more electrons. Okay, fine. So reduction. Something has to gain those electrons. This is a classic oxidation reduction reaction. We're burning candles, okay? If we were on the ground, I'd actually be doing this. In an oxidation reduction reaction, right here, we are stripping away electrons from the wax of the candle. And that energy is being released as heat and light. We're also giving off carbon dioxide and water because we're using up oxygen. Now, if we were to take a glass container and place it over the candles burning here in this picture, in this animation, if we were to take a big beaker and put that over the candles would go out. And most people would say it went out because it used up all the oxygen gone. But that's not true. What happens is when you put, if you have like one of the candles that comes with a lid, like a Yankee candle, or something at Walmart that has a bunch of candles that look like Yankee candles, and you put the lid on it, the flame goes out. It doesn't go out because of the lack of oxygen. The burning of a candle releases carbon dioxide and water. If you were to put a glass over a glass container, like a beaker, over these candles, the candles would go out. but you'd also see a ring of moisture condensing on the inside of the beaker. And that tells you that water vapor is coming off it. So we know that water vapor is being produced in the break, we know that oxygen is accepting electrons and hydrogens because water is forming. We don't see it until we, put a beaker over top of it. We also know that we haven't used up the oxygen. We've generated more carbon dioxide. Carbon dioxide is heavier than air. If we take a beaker and cover those candles, we know that carbon dioxide is going to be there because the candles will go out because it displaces the oxygen. The oxygen has risen to the top of the beaker. You could stick a glowing wick, you could stick a match that is, still glowing up. If you could take the beaker and lift it up and stick it up into the upside, never mind. Take my word for it. If you put a beaker over top of this, you're going to have carbon dioxide at the bottom where the candles are, and that will displace the oxygen. The oxygen is lighter than the carbon dioxide, and the oxygen will rise to the top of the beaker and be trapped there. Example, when we used to do science camp for middle school in the summer, we used to take a Coke bottle, an empty Coke bottle, and we would put vinegar in the Coke bottle. And you may have done this too. You take vinegar in the Coke bottle, take a balloon and fill the balloon full of baking soda, and then put that on the top of the Coke bottle, tilt the balloon up and dump all the baking soda into the vinegar, and you're going to release a lot of carbon dioxide, which is going to rise up and inflate the balloon. If you take the balloon off, which is now filled with carbon dioxide, tie it off, and then bounce it, it's going to drop to the floor because the balloon is filled with carbon dioxide. It's heavier than air. So we know that carbon dioxide is present. Just like here, we know carbon dioxide is present. How do we know it's present in our bodies? Because we're breathing it out. Okay. This whole process of cellular respiration is a very controlled release. We don't want to rip off all the electrons. We don't want to rip off all the hydrogen atoms in one explosive step. I mean, consider this. If we did that, if we released all the energy in those candles at once, they would flash and probably explode. It'd be very exciting, very challenging for a birthday cake, because the candles would explode. Because that's what would happen if we didn't have a step-by-step process. So we have in our bodies, we have a gradual breakdown. Some oxidation reactions are very rapid. When you light a match, that's a fairly rapid oxidation. It's not explosive, but it's fairly rapid. If you light a firecracker, that's a fairly rapid oxidation. You burn gasoline or diesel in your engine, that's a fairly rapid oxidation. You leave your shovel out all winter and it rusts and falls apart, that's still oxidation. It's just slower. Again, it depends on the presence of oxygen. The metal is going to rust because oxygen is going to be accepting the electrons coming off of the metal. And you're going to get this iron oxide covering that's going to be weaker and weaker. And then in the spring, you got to dig your first hole in the ground and the shovel breaks apart. The rate of oxidation it can be very rapid, it can be very slow, but it's still the same process. The rusting shovel and the burning match is still oxidation. The only difference is the speed with which it operates. We want to have a nice gradual breakdown in our bodies because we don't want to have an explosion. We use our enzymes to gradually strip away the hydrogen ions and the electrons from the glucose, and we transfer them to these intermediate molecules that get reduced. We don't bring in oxygen to play until we're near the end of the whole sequence. We want to have a controlled release. We don't want to have this. This is a real-time video of the Hindenburg exploding back in the 1930s. The Hindenburg was a Zeppelin. It was filled with giant gas bags filled with hydrogen, elemental hydrogen, very reactive, looking for its electrons and wanting to stabilize its valence shell. Well, it encountered oxygen and produced a lot of water in this process. It also released a lot of energy all at once. No intermediate steps here. We went from there 1 second and releasing energy the next second. So we don't want that in our bodies. To make the oxidation reduction reactions work in our bodies, we have to use intermediate steps. We use coenzymes. Remember, vitamins act as coenzymes for enzymes. We also have coenzymes that work with enzymes to function as electron and hydrogen acceptors. Now, the ultimate acceptor of an electron is always going to be oxygen. That's the classic textbook answer. The ultimate electron acceptor in the human body is oxygen. Always. Oxygen will always take electrons. But we got to control how we can feed those electrons to the oxygen. So in breaking down glucose, we have to do it slowly. We have to go step by step. We can't use oxygen until we get to the very end. So we have to use these intermediate products as acceptors of electrons. They get temporarily reduced. We have these hydrogen acceptors, the electron acceptors. They're called NAD+, nicotinamide adenine dinucleotide, which is why we call it NAD+. Plus, because in its unreduced state, it has a positive charge. The other one is flavin adenine dinucleotide, or FAD. FAD and NAD+ are the two hydrogen/electron acceptors we use as intermediate carriers, because we don't use oxygen to accept electrons until the very end of cellular respiration. Now, this process of aerobic cellular respiration is a redox reaction. It's an oxidation reduction reaction. We take the hydrogens off the glucose with their electrons, and we bring them to oxygen. So we're simply transferring hydrogen ions and the high energy electron that goes with them. And whatever accepts that hydrogen and the electron gets reduced. So whatever we're going to make is going to get reduced. You know, when NAD+ accepts a hydrogen ion and its electron, it's gotten reduced. Now, these electrons, I can't emphasize this enough again. The electrons that we're talking about are the valence electrons between the carbons and the hydrogens in our glucose molecules. Lots of potential energy there. We strip away the hydrogens and the electrons, and that energy is transferred to something else. We use that energy. The ultimate goal is to use that energy that we're pulling off the electrons, pulling off the glucose, to make ATP. We can't take the energy directly from glucose and apply it to a muscle cell. We can't apply it to a nerve cell. We can't apply it to the sodium-potassium pump directly. We have to go through the production of ATP, because ATP is what we use to get that energy to the nerve cell, to the muscle cell, to the sodium-potassium pump. The energy is contained the energy from the high energy from the electrons is temporarily parked in the ATP molecule. And it's the ATP molecule that we use to transfer the energy to the pump, to the muscle cell, to the nerve cell. Okay, so we know all this. So the electrons in the valence shell are unstable because they have more potential energy. Stable electrons are in close. They're in the first shell, the closer orbits. The outer shell, the outer orbit, is where the unstable electrons are because they got lots of energy to give. And we can use this energy when we break those valence bonds to make ATP. And that's why we love carbohydrates and fats, because they have lots and lots and lots of carbon and hydrogen bonds. Now the formula for all this is very simple. C6H12O6 plus 6O2, meaning 1 glucose molecule, C6H12O6, and 6 oxygens will yield always 6 CO2s and 6 waters, and the release of energy as ATP and also as heat. Glucose gets oxidized to carbon dioxide. Glucose gives up all 12 of its hydrogen atoms. And the end result of doing that causes the formation of carbon dioxide. Oxygen gets reduced to water. Oxygen grabs all those hydrogens. And we end up making 6 water molecules. Those 12 hydrogen atoms, two hydrogens to an oxygen, we make H2O, and we get 6 water molecules. So the end result of oxidation of glucose is C6H12O6 in the presence of oxygen, in the presence of 6 oxygen atoms, 6 oxygen molecules, will give you 6 carbon dioxide molecules, 6 water molecules, and energy in the form of ATP and heat. Now, the enzymes that we use here to make this work are known as dehydrogenase enzymes. Remember I said hydrogenation adds hydrogens to a molecule, like, hydrogens to an oil makes it a solid. Dehydrogenase enzymes strip hydrogens away. We take the hydrogens off of the glucose, and we take their electrons with it. So the dehydrogenase enzymes take the glucose, strip off the electrons, strip off the hydrogens. That energy can be transferred directly to ATP, but usually the energy, when it's stripped off, is transferred to the electron acceptor, which is the NAD+. Because remember, electrons have to go somewhere. They can't just run randomly through the air and keep bouncing around. When electrons are released through oxidation, they have to be accepted somewhere else. And that somewhere else is usually oxygen. But when we're trying to work our way down to the oxygen, we have to use an intermediate electron acceptor. And that's where the NAD+ comes in. So the dehydrogenase enzymes strip off the hydrogens and the electrons from the glucose, and the NAD+ grabs those hydrogens and electrons and becomes NADH+, H+. Protons are hydrogens. They are a positively charged hydrogen atom. They're a hydrogen ion. That's what a proton is. And every time a hydrogen comes off, an electron goes with it because they keep the charges balanced. Yes, the test dates have been moved back. So, and that's, I may change those again, but we'll see. So, but right now everything is set for the 22nd. Lab test is set for the 22nd. That's not going to change. Lecture test, I may push that back. So, but yes. Okay, now, food contains lots of high energy bonds. And those high energy bonds are the high energy electrons. The electrons in the valence shells are what we call high energy electrons. They have lots of potential energy. And they've got their hydrogen atoms with them, their hydrogen ions with them. So we have lots of hydrogen atoms bonding with carbon, lots of high energy electrons. And we want to use, we can use those high energy electrons to do things. So, so we eat a Snickers bar, we eat an apple, we eat a banana, we eat a carrot, we eat a Whopper, or a quarter pounder, or we eat steak or chicken, or drink a glass of milk, or eat ice cream or something, anything that has energy in it. So all of our food contains high energy bonds with high energy electrons. And here between the carbons and the hydrogens, there are lots of high energy electrons. And we need those hydrogens and we need those electrons. And we will transfer the electrons to the NAD plus as an intermediate step as we start stripping away electrons and hydrogens from glucose. The whole process of oxidation involves stripping away electrons and stripping away hydrogens. As we do that, we have to have this intermediate carrier to grab the electrons and grab the hydrogens, because we're not taking them directly to oxygen. We don't want to go that fast. That's a little faster than our bodies would like. So we have this intermediate step. Let's use the NAD pluses, for example, as to slow the oxidation of glucose down. It isn't like burning, wood in a fire pit. We want it to be a little bit slower here. So we use our NAD plus. We don't get to the use of oxygen until we get to the electron transport chain. Now, every ATP molecule will last for about 7 seconds. And then we have to make another one. It only lasts, we can't store ATP. We keep making it each one will last for about 7 seconds. Each ATP molecule contains about 7.3 kilocalories of energy. So for 7 seconds, we've got that energy. Then we have to make more, we have to make more, we have to keep making more. Now there's 2 ways to make ATP. What is called substrate level phosphorylation. And that only occurs in glycolysis. Substrate level phosphorylation. Now don't get put off by the title. Take it apart. What does substrate mean? Substrates are the things that go into the active sites of the enzymes. Substrate level phosphorylation. Make sure you know this. Make sure you own this slide, because you'll see this question again. Substrate level phosphorylation involves the use of enzymes to generate ATP. These dehydrogenase enzymes take the hydrogens and the high energy electrons off of the glucose and use that to push a phosphate back onto push the third phosphate onto ADP to make the high energy compound ATP. Because ATP is a very simple process. You have the adenosine diphosphate with the 2 phosphates on the end, and you have the third phosphate being brought in every 7 seconds with energy. And so the third phosphate is put in there in a high energy bond. And that lasts for 7 seconds, and then the bond breaks, and then we have to do it again and again and again. And the energy for that bond in ATP comes from 2 ways. The simplest approach is the substrate level phosphorylation. We drop the glucose molecule into the active sites of the dehydrogenase. It strips off the hydrogens, it strips off the electrons. The energy from that is used to form ATP. It's how we produce ATP in glycolysis. Substrate level phosphorylation. It's the use of the enzyme. We use the glucose as the substrate on the active site, just like amylase did in our mouths for starch. Here we're using glucose inside the cytoplasm to rip off the hydrogens and the electrons. That's the simple way to generate ATP, but it doesn't use oxygen. It doesn't make very much ATP. The other way takes place inside the mitochondria. This is known as oxidative phosphorylation. We call it that because we use oxygen to drive the phosphorylation. We use oxygen to receive the electrons when they're at the end of their run. We use the energy from the electrons to pump hydrogens into this area between the two, in the area called the inner membrane space. We use the electrons to pump the hydrogen. The hydrogens is going to diffuse back into the matrix of the mitochondria. And in doing so, it runs through this device called an ATP Synthase enzyme, and it makes ATP. And it sounds like it's all magic, doesn't it? Well, there's no such thing as magic, but that's exactly how it works. Anyway, what you need to remember from this slide is oxidative phosphorylation is used in electron transport. Now, is that ever going to be a question you're going to get asked in a clinical setting? Very unlikely. Very unlikely. But there is a reason why we talk about this. I'm going to get to that. This is substrate level phosphorylation. The purple things are the enzymes. They're not really purple, but they are here. Purple things are the enzymes. The substrates land on the enzyme. The substrates are the glucose molecule. The enzyme is the decarboxylase enzyme. The dehydrogenase enzyme that's pulling off the hydrogens and the electrons. The energy from the electrons is transferred to the ADP here to force the third phosphate back on. And that makes ATP. That's all ATP is. It is the ADP and inorganic phosphate shoved together in this high energy bond. And we get the energy from glucose to make the ATP. And we have we don't have a lot of ATP being made this way, but we have some. We don't use oxygen in here. When we break apart when the dehydrogenase breaks apart the glucose, those electrons and those hydrogens end up going to NAD+. Because NAD+ is getting reduced, we still have to have reduction. We have oxidation, we have to have reduction. We don't make a lot of ATP, we make some. And it takes place in glycolysis, the breakdown of glucose in the cytoplasm of the cell. The first step in the whole sequence of events is glycolysis in the cytoplasm. We take glucose and start breaking it down. And we use these enzymes, these dehydrogenase enzymes to do so. And we get a little bit of ATP being formed. And we have actually, but more importantly, we're parking electrons and hydrogens in the NAD pluses. So oxidative phosphorylation takes place in the mitochondria. Here we are going to use a series of enzymes embedded in the inner membrane of the mitochondria. It has an inner membrane and an outer membrane. And these electron transport enzymes are embedded in the inner membrane, and they will pump, they function as pumps. They pump hydrogens from the NAD, the NAD plus captured a bunch of hydrogens and electrons. These enzyme pumps will pump the hydrogens across the inner membrane into the space between the inner and outer membrane using the power of the electrons. And it will pump all these hydrogens into that space. Now those hydrogens want to reach equilibrium with the hydrogens in the matrix, the middle of the mitochondria. But there's no way for them to get through the membrane, except there is one enzyme called the ATP synthase. And the hydrogens can move through that synthase, but it will cause the synthase to mechanically turn. And that rotating action of the synthase forces phosphates onto ADP molecules, and that energy formed, the energy captured in the mechanical process gives us the high energy bond at the third phosphate. And now we have a whole bunch of ATPs. This is what the ATP synthase looks like. It's an enzyme. It's embedded in the membrane. The food energy that we have in our glucose is, of course, captured is found in the high energy electrons. Those electrons are used by these pumps, these enzyme pumps, to pump hydrogens across this membrane. We call them proton pumps, because that's what they're pumping. The hydrogens are protons. We're pumping the hydrogens into this space here. We have billions of hydrogens out there and maybe millions down here. The hydrogens want to reach equilibrium. And so the only way to get back into the matrix in the middle of the mitochondria is to pass through the ATP synthase. And as it does, it causes this structure here to turn and mechanically force those inorganic phosphates and ADP together, creating the high energy ATP. The energy of turning, the energy of the hydrogens moving through here causes us to rotate and park energy here, and we get a whole bunch of ATPs. And this is oxidative phosphorylation. And it's a whole lot more complicated than substrate level. Now, why do we care? Well, what happens when we run out of hydrogens? Or what happens when we run out of electrons? Or what happens when we don't have oxygen to accept all these electrons coming through here? When these electrons that are used to pump the hydrogens across use up their energy, they drop back down here and they're grabbed by oxygen. And those electrons and their hydrogens form water. What happens if there's no oxygen present? The hydrogens don't have anywhere to go. And the hydrogens start accumulating in the matrix. Pretty soon you reach equilibrium and the production of ATP stops. Now that, this is what happens when our cells run out of oxygen, when our cells start to die. It's because of we have no place for the hydrogens to go and no place for the electrons to go. So, and if something blocks the ability of high oxygen to be there, like a toxin or a poison, the cell dies too. So, okay. So this is a great place to stop. I'm going to stop here. And I've given you a lot to think about on this. We will pick up with this again on Tuesday and wrap up this chapter Tuesday. And I plan to go into start the bone chapter on the physiology of the bones on Tuesday, because there aren't many more slides left to address here. So, and we'll go from there. I'll take a look at the date. I thought we might get done today on this chapter. And so I'll probably slide the lecture test date back a little bit by a couple days, just to make sure you have plenty of time to prepare. So, and yeah, everything in Unit 2, in the content section, all the information on the cells and protein synthesis in particular, and then you know, the information on metabolism as far as nutrition and then metabolism. You know, so there are, I guess there are you know, 4 key areas to look at. You know, cellular respiration, the nutrients, protein synthesis, and then the cell structure. So those are the 4 key substances, but everything you need is in the content section. The study guides, lecture study guides, I think are I think they're pretty good. I think they'll help you a lot. So, anyway, we're going to wrap up. We'll wrap up this on Tuesday and go from there. A reminder that there is no lab this afternoon, since we didn't have any labs this week at all, because we lost 2 lab days by fall break. So there's, you know, I didn't want to get out of sequence. So we'll start up with our labs again next week, and we'll be looking at muscles. So, okay, unless anybody has any other questions, I'm going to get us out of here, and I'll see you all next week.